Section 1. What is agrobacterium-mediated gene transfer? Agrobacterium-mediated gene transfer is a biotechnological technique that uses soil bacteria to insert foreign genes into plant cells. This method has become one of the most widely used approaches for creating genetically modified plants. The technique uses Agrobacterium tumefaciens, a soil bacterium that naturally infects plant wound sites. What makes this bacterium special is its natural ability to transfer a segment of DNA, called tDNA, from its tumor-inducing plasmid into the plant's genome. Let's explore how this process works and why it's so important for plant biotechnology. To understand agrobacterium-mediated gene transfer, we need to compare the natural process with its biotechnological application. In nature, agrobacterium transfers tDNA containing genes that alter plant hormone production, causing tumorous growths known as crown gall disease. Scientists have ingeniously modified this system by replacing the tumor-inducing genes with genes of interest, creating a powerful tool for plant genetic engineering. Agrobacterium is a soil-dwelling bacterium that has evolved a remarkable ability to genetically modify plants. In nature, agrobacterium targets plant wound sites, exploiting the plant's vulnerability to infection. The bacteria are attracted to chemical signals released by wounded plant tissue. Once at the wound site, agrobacterium transfers a portion of its DNA into the plant cells, causing crown gall disease, characterized by tumor-like growths. The transferred genes force the plant to produce unique compounds called opines. These serve as specialized nutrients that only the agrobacterium can use, creating a parasitic relationship. Scientists recognize the remarkable potential of agrobacterium's natural gene transfer ability and modified it for biotechnology applications. By removing the disease-causing genes while preserving the DNA transfer mechanism, researchers transformed this plant pathogen into a powerful tool for genetic engineering. This ingenious repurposing allows scientists to insert beneficial genes into plant genomes with precision without causing disease. Essentially, nature's genetic engineer has been harnessed for sustainable crop improvement. Agrobacterium is a genus of soil bacteria widely used for plant genetic transformation. There are three main Agrobacterium species used in plant biotechnology, each with unique characteristics. Agrobacterium tumefaciens is the most commonly used species. It causes crown gall disease in plants, contains the Ti plasmid, and can infect a wide range of plant species. Agrobacterium rhizogenes induces hairy root disease. It contains the root-inducing or Ri plasmid and is particularly useful for generating transgenic root cultures. Agrobacterium vitis is specific to grapevines, causing grape crown gall disease. It has a narrower host range and is primarily used in viticulture applications. Let's explore how each species is applied in plant biotechnology. Agrobacterium tumefaciens is widely used for crop improvement, including introducing herbicide resistance, insect resistance, and enhancing nutritional qualities. Agrobacterium rhizogenes is particularly valuable for producing secondary metabolites and medicinal compounds, studying root systems, and developing stress-tolerant plants. Agrobacterium vitis is applied specifically in grapevine improvement, enhancing disease resistance, wine quality, and drought tolerance in vineyards. When choosing an agrobacterium species for plant transformation, consider these guidelines. Choose Agrobacterium tumefaciens for most dicot crops and general applications. Use Agrobacterium rhizogenes when root development or root cultures are desired. Select Agrobacterium vitis specifically for grapevine transformation. Always consider bacterial strain compatibility with your plant host and match your transformation goals with the specific plasmid properties of each species. Each agrobacterium species offers unique advantages for specific plant transformation objectives. Understanding their differences is crucial for successful genetic engineering in plants. The Ti plasmid, or tumor-inducing plasmid, is a crucial component in agrobacterium-mediated gene transfer. This circular DNA molecule contains several key regions that work together during the infection and genetic transformation process. The tDNA, or transferred DNA, is the region that gets integrated into the plant genome. In nature, this region contains genes that induce tumor formation and opine synthesis. The virulence genes, or vir genes, encode proteins that recognize plant signals and facilitate the transfer of tDNA into plant cells. The opine catabolism genes allow Agrobacterium to metabolize opines produced by the transformed plant cells, providing a selective advantage. The origin of replication ensures that the plasmid can replicate independently within the bacterial cell. The tDNA is flanked by specific 25 base pair sequences called border sequences. These borders are essential for tDNA transfer, as they are recognized by the vir proteins during the transfer process. For biotechnology applications, the TI plasmid is modified by removing the tumor-inducing genes from the tDNA region. Scientists replace these genes with the gene of interest they want to introduce into plants, while keeping the essential border sequences intact. The virulence genes are maintained as they are essential for the DNA transfer mechanism, and the origin of replication is preserved to ensure plasmid survival in bacteria. To summarize, the key points about TI plasmid modification include keeping the border sequences intact, removing tumor-inducing genes, preserving virulence genes, and inserting the gene of interest within the tDNA region. This understanding of the TI plasmid structure is essential for developing binary vector systems, which we'll explore next. Binary vector systems revolutionized plant genetic engineering by separating the Agrobacterium TI plasmid into two distinct components. The traditional TI plasmid system contains both virulence genes and the tDNA region on a single large plasmid. Scientists developed the binary vector system to overcome the challenges of manipulating the large TI plasmid. In this improved system, the virulence genes are carried on a helper plasmid, which is a disarmed TI plasmid with the tDNA region removed. The tDNA region is then placed on a separate, smaller binary vector, which is much easier to manipulate in the laboratory. This binary vector contains the tDNA borders and the gene of interest that researchers want to introduce into the plant genome. The binary vector system offers several significant advantages for plant genetic engineering. First, it makes manipulating the tDNA region much easier, as researchers work with a smaller plasmid. The smaller size of the binary vector also makes cloning procedures more efficient and reliable. This system provides higher transformation efficiency compared to traditional methods. Finally, it offers greater flexibility in designing and testing different genes and constructs. Let's examine how this binary vector system works at the molecular level. The process begins when virulence genes on the helper plasmid sense signals from wounded plant cells. These activated virulence proteins then process the tDNA region from the binary vector, preparing it for transfer. Next, the tDNA containing the gene of interest is transferred from the bacteria into the plant cell. Finally, the tDNA integrates into the plant genome, allowing expression of the introduced gene. By separating these components, scientists can more easily engineer plants with new traits while maintaining efficient transformation. For successful agrobacterium-mediated transformation, specific laboratory equipment is essential. Sterilization equipment like autoclaves operating at 121 degrees Celsius and 15 psi are essential for creating sterile materials. These devices eliminate microbial contaminants that would interfere with transformation. Laminar flow hoods and biosafety cabinets provide sterile working environments by creating a positive pressure of filtered air. These workspaces are critical for handling plant tissues and bacterial cultures. Specialized growth chambers with controlled temperature, humidity, and light cycles are needed to maintain optimal conditions for both bacteria and plant tissues during transformation. Equipment like centrifuges for bacterial culture preparation and microscopes for examining transformed tissues are essential for monitoring and analyzing the transformation process. Contamination is the primary cause of failed transformations. Even minor lapses in sterility can introduce microorganisms that compete with Agrobacterium or cause tissue damage, requiring strict adherence to aseptic techniques. To summarize, proper equipment and maintaining sterile conditions are critical for successful agrobacterium-mediated transformation. This section covers the essential materials and reagents required for agrobacterium-mediated gene transfer. Plant tissue culture media forms the foundation for growing plant tissues. Common formulations include Murashige and Skoog medium and Gamborg's B5 medium, which provide essential macro and micronutrients, vitamins, and carbon sources for plant growth. Selection antibiotics are crucial for identifying transformed plant cells. Antibiotics like kanamycin and hygromycin select for plant cells containing the resistance gene, while carbonicillin and cefotaxime help eliminate residual agrobacterium after transformation. Plant growth regulators control cell division and differentiation. Auxins like 2,4-D and NAA promote callus formation and root development, while cytokinins such as BAP and zidin stimulate shoot development. The balance between these hormones determines regeneration outcomes. Sterile culture containers are essential for maintaining aseptic conditions. Petri dishes, culture tubes, and specialized plant culture vessels provide the environment for plant growth. Proper sealing with micropore tape or parafilm ensures sterility while allowing gas exchange. Bacterial culture media are used for growing agrobacterium strains. LB, YEB, and MGL media provide nutrients for bacterial growth. These media are supplemented with antibiotics to maintain the selection pressure for the binary vector system. Quality and sterility are non-negotiable in agrobacterium-mediated transformation. Contamination with fungi or bacteria can lead to complete experimental failure. All media must be prepared with high-grade reagents and properly sterilized by autoclaving or filter sterilization for heat-sensitive components. To ensure successful agrobacterium-mediated transformation, maintain a carefully prepared inventory of all essential materials including media, antibiotics, growth regulators, sterile containers, and bacterial culture supplies. With these materials prepared, we are ready to proceed to the next steps in the transformation process. This section covers seed sterilization and germination, essential steps in agrobacterium-mediated transformation. The process involves 5 critical steps: seed selection, surface sterilization, washing, plating on sterile media, and controlled germination. Begin by selecting high-quality, mature seeds of uniform size. This ensures consistent results and reduces contamination risks. Several sterilization methods can be used. Sodium hypochlorite at 1 to 5% is common for 5 to 15 minutes. 70% ethanol provides rapid sterilization, but only for 30 to 60 seconds to avoid seed damage. Hydrogen peroxide is a gentler alternative. It's critical to note that extended exposure to sterilizing agents can damage seeds and reduce germination rates. A typical protocol begins with a brief ethanol rinse, followed by sodium hypochlorite treatment for 10 to 15 minutes. Seeds must then be thoroughly rinsed with sterile distilled water, typically 3 to 5 times. The washing process is critical for removing all traces of sterilizing agents. Each wash involves gently agitating the seeds in sterile water. After sterilization, seeds are placed on sterile growth medium containing essential nutrients, a carbon source like sucrose, and a solidifying agent such as agar, with pH adjusted to 5.7 to 5.8. Germination occurs under carefully controlled conditions: temperature of 22 to 25 degrees Celsius, 16-hour light and 8-hour dark photoperiod, moderate light intensity, and controlled humidity. Most plant species begin germination within 3 to 4 days, as evidenced by radical emergence. By 7 to 10 days, seedlings develop true leaves and are ready for transformation experiments. Remember these key points. Start with high-quality seeds. Balance sterilization efficacy with seed viability. Ensure thorough washing. Maintain aseptic conditions. And recognize that healthy seedlings are essential for successful transformation. With properly sterilized and germinated seedlings, we can proceed to the next step: preparing the agrobacterium culture for transformation. Preparing agrobacterium cultures is a critical step for successful plant transformation. First, select a single colony of agrobacterium containing your gene of interest. Inoculate it into liquid media such as LB or YEB, supplemented with appropriate antibiotics for selection. Incubate the inoculated culture at 28 degrees Celsius with shaking at 180 to 200 RPM. Monitor bacterial growth by measuring optical density at 600 nanometers. The optimal density for transformation is between 0.6 and 0.8, which typically occurs during the log phase of bacterial growth. Once the culture reaches optimal density, transfer it to sterile centrifuge tubes. Centrifuge at 3,000 to 5,000 grams for 10 minutes to pellet the bacterial cells. After centrifugation, carefully discard the supernatant, leaving the bacterial pellet intact. Resuspend the bacterial pellet in fresh liquid medium. Adjust the concentration to OD 600 of 0.3 to 0.5 for optimal transformation efficiency. Add acetylsyringone to induce virulence genes. Allow the suspension to incubate for 1 to 3 hours before using it to infect plant tissue. To maximize transformation efficiency, use fresh bacterial cultures that are 18 to 24 hours old. Ensure precise optical density measurements and maintain sterile conditions throughout the process. For optimal results, adjust the protocol based on the specific plant species being transformed. Explant preparation is a critical step in agrobacterium-mediated transformation. Explants are plant tissue segments that are used for transformation and the regeneration of transgenic plants. Different types of explants can be used depending on the plant species and experimental goals. Common explant types include leaf discs, cotyledons, hypocotyls, and stem segments. Explants must be carefully prepared to create wound sites. For leaf discs, a cork borer is commonly used to cut uniform circles from intact leaves. Wounding is essential for successful transformation. Cut edges release phenolic compounds that attract agrobacterium and create entry points for bacterial invasion. The choice of explant varies significantly by plant species. Some plants, like tobacco, transform efficiently using leaf discs, while others may require specialized tissue, like embryogenic callus. Multiple factors affect transformation success rates. The age of donor tissue and size of explants are particularly important, with young tissues and smaller explants generally yielding better results. When preparing explants, maintain strict sterile conditions to prevent contamination. Use young, actively growing tissue and create clean, precise cuts to optimize transformation efficiency. Proper explant preparation is a foundation for successful agrobacterium-mediated transformation and can significantly impact your experimental outcomes. Section 11. Infection and co-cultivation. The infection process begins by immersing plant explants in an agrobacterium suspension. The bacteria surround and attach to the plant tissue, initiating the gene transfer process. After infection, the explants are transferred to co-cultivation media without any selection agents. Co-cultivation typically lasts for 2 to 3 days. During this critical period, agrobacterium transfers its tDNA into the plant cells. At the molecular level, agrobacterium attaches to the plant cell and creates a channel to transfer its tDNA. Controlling co-cultivation conditions is crucial for successful transformation. Important factors include temperature, light, pH, and hormone balance. It's essential to balance transformation efficiency with bacterial control. Extended co-cultivation can lead to bacterial overgrowth, reducing transformation success. After co-cultivation of plant tissue with agrobacterium, washing is a critical step. Excess bacteria must be removed to prevent overgrowth that could damage or kill the plant tissue. Proper washing protects the explant's viability while ensuring that transformation has had sufficient time to occur. Let's examine the step-by-step washing procedure to eliminate excess agrobacterium from plant tissues. First, prepare sterile wash solutions and antibiotics at appropriate concentrations. Next, carefully transfer the co-cultivated explants to a sterile container to begin washing. Wash the explants 3 to 4 times with sterile distilled water to remove the majority of bacteria. In the final wash, add appropriate antibiotics to kill any remaining bacteria. Finally, transfer the explants to selection media containing antibiotics to continue the transformation process. Several antibiotics can be used to eliminate agrobacterium. Here are the most commonly used options and their typical concentrations. Cefotaxime is commonly used at concentrations between 250 and 500 milligrams per liter to inhibit bacterial cell wall synthesis. Timentin, a mixture of ticarcillin and clavulanic acid, provides broad-spectrum activity at 150 to 300 milligrams per liter. Carbonyclin serves as an alternative to cefotaxime and is used at similar concentrations. Now let's visualize how the bacterial washing process works. Initially, the explant is surrounded by a high density of agrobacterium cells after co-cultivation. After the first wash with sterile water, we see a significant reduction in bacterial numbers. The second wash further reduces bacterial density. Finally, after treatment with antibiotics, virtually all bacteria are eliminated while the transformed plant DNA remains integrated. There are several critical considerations for successful bacterial elimination. Timing is crucial. Allow 24 to 48 hours of co-cultivation before washing to ensure proper gene transfer. Wash solutions should be at room temperature to minimize stress to the plant tissues. Gentle handling of explants is essential to minimize tissue damage during the washing process. Maintain strict aseptic conditions throughout to prevent contamination. Select antibiotics based on the specific agrobacterium strain used for transformation. After plant tissues have been infected with agrobacterium and co-cultivated, the next critical step is selecting cells that have successfully incorporated the transferred DNA. The selection process relies on antibiotic or herbicide resistance genes that are transferred alongside the genes of interest. These resistance genes allow transformed cells to survive in the presence of selection agents. When selection agents are added to the culture medium, non-transformed cells cannot survive because they lack the resistance gene. In contrast, transformed cells express the resistance gene and can detoxify or block the selection agent, allowing them to grow and divide. Several selection agents are commonly used in plant transformation. Each works with a specific resistance gene that counteracts the agent's toxic effects. The mechanism of resistance varies depending on the selection system used. In most cases, the resistance gene encodes an enzyme that inactivates the antibiotic or herbicide. The selection process isn't a one-time event. Selection pressure must be maintained through multiple subcultures over several weeks to ensure only truly transformed cells continue to grow. With each subculture, the proportion of transformed cells increases as any remaining non-transformed cells are eliminated. This process typically takes 3 to 4 weeks. To summarize the key points about selection, selection agents eliminate non-transformed cells while transformed cells with resistance genes survive. Multiple subcultures ensure pure transformance, and selection pressure must be maintained throughout the process. After successful transformation and selection, the next critical step is shoot regeneration. This begins with transformed callus tissue, which consists of undifferentiated cells. Shoot regeneration relies heavily on plant hormones called cytokinins. These specialized hormones, including BAP, TDZ, kinetin, and zeatin, stimulate cell division and promote shoot formation from the callus. The media formulation is critical for successful shoot regeneration. It typically contains MS or B5 basal salts, vitamins, a carbon source like sucrose, and most importantly, cytokinins at concentrations between 0.5 and 5 milligrams per liter. The ratio of cytokinins to auxins must favor cytokinins for shoot development. The media also includes selective agents to maintain selection pressure and prevent growth of non-transformed cells. Shoot regeneration is a time-intensive process that typically unfolds over several weeks. After transferring the callus to regeneration media, the first signs of regeneration appear within 1 to 2 weeks as small green spots indicating chlorophyll production. By weeks 3 to 4, shoot primordia begin to form as organized meristematic centers. During weeks 5 to 6, these primordia elongate into recognizable shoots, and by weeks 7 to 8, leaf development becomes apparent. The visual progression of shoot regeneration begins with small green spots on the callus, indicating areas where cells are beginning to organize into meristematic regions. These develop into shoot primordia, which are small, dome-shaped structures with organized cell layers. The early shoots then begin to elongate with rudimentary leaf structures. Finally, fully developed shoots emerge with well-formed stems and leaves, ready for the root induction phase. Several factors critically influence shoot regeneration success. The hormone concentration and ratio is particularly important, as is the explant source and age. Environmental conditions like light intensity and temperature must be carefully controlled. When troubleshooting regeneration issues, consider trying different cytokinin types and concentrations. If shoots develop abnormally with a glassy appearance, this vitrification can be addressed by reducing humidity in culture vessels. Slow growth may indicate suboptimal light quality or temperature. Successful shoot regeneration is indicated by multiple healthy green shoots with normal leaf morphology that continue to grow on selective media. Once shoots reach 2 to 3 centimeters in height and have developed several leaves, they're ready to move to the next stage, root induction. Root induction is a critical phase in plant transformation where regenerated shoots develop root systems to become complete plantlets. During this phase, regenerated shoots are transferred from shoot induction media to specialized rooting media. This transfer must be done under aseptic conditions to prevent contamination. Auxins are the key plant hormones responsible for root development. Common auxins used include IAA, NAA, and IBA, which stimulate cell division and differentiation in the stem base. When exposed to the correct auxin concentration, cells at the base of the shoot begin to divide and differentiate into root primordia, eventually forming a complete root system. The efficiency of root induction varies greatly between plant species. Some plants, like tobacco or tomato, develop roots easily with standard protocols. Other species, particularly woody plants and some cereals, are more recalcitrant to rooting and may require higher auxin concentrations or additional treatments to stimulate root development. For difficult-to-root species, several additional treatments may be employed. These include pulse treatments with high auxin concentrations, the addition of phenolic compounds, or physical wounding of the stem base to stimulate rooting. Other factors that can enhance rooting include optimizing light and dark exposure periods, adjusting temperature, or adding specific rooting cofactors depending on the plant species. The end result of successful root induction is a complete plantlet with a functional root system connected to the shoot. These roots will allow the plant to absorb water and nutrients independently. A well-rooted plantlet represents the final stage before acclimatization to soil conditions, bridging the gap between in vitro and in vivo environments. After successfully inducing roots, the complete plantlets are ready for the next critical phase, acclimatization to soil conditions. Acclimatization to soil is a critical step in transferring transformed plantlets from tissue culture to normal growing conditions. The process begins with carefully removing plantlets from their sterile culture vessels. These plantlets must be gradually adapted to soil conditions, as they've been growing in a protected, high-humidity environment. The acclimatization process consists of several critical steps. First, carefully transfer plantlets to a sterile potting mix with good drainage. Second, cover the plants with a humidity dome or plastic bag to maintain high humidity. Third, gradually reduce humidity by incrementally opening vents or lifting the dome over 1 to 2 weeks. Fourth, provide controlled light exposure, starting with indirect light and slowly increasing intensity. Finally, transition to a normal fertilizer regime as plants establish. The most critical aspect of acclimatization is the gradual humidity reduction. Initially, plants are kept at nearly 100% humidity using a dome or plastic covering. After about a week, begin creating small openings to gradually reduce humidity. By the end of the second week, the cover can be removed completely as plants adapt to ambient humidity. Several key factors determine successful acclimatization of transformed plantlets. First, timing is critical. Transfer plants during their active growth phase. Temperature should be maintained between 22 and 25 degrees Celsius. Light exposure should start with indirect light and increase gradually. The substrate should be a well-draining sterile mix, often containing perlite. Regular monitoring is essential to identify and address any signs of stress. Proper acclimatization ensures plants can successfully transition from controlled lab conditions to natural environments. During this process, plants shift from heterotrophic to fully autotrophic growth. Functional stomata develop for normal gas exchange. Plants form a protective cuticle layer to prevent water loss. The root system strengthens for efficient nutrient uptake from soil. All these adaptations enable the transformed plants to establish successfully in natural growing conditions. After the plant transformation process, confirming successful gene integration is a critical step. Scientists use 4 key methods to verify that the genetic transformation was successful. PCR, or polymerase chain reaction, detects the presence of the inserted gene in the plant DNA. This technique uses specific primers that bind to and amplify the transgene sequence if it's present, providing a quick and reliable first confirmation. RT-PCR, or reverse transcription PCR, goes beyond detection to verify if the transgene is being expressed at the RNA level. This method first converts messenger RNA to complementary DNA, then performs PCR, indicating whether the gene is functionally active in the plant. Southern blotting is a more definitive technique that confirms actual integration of the gene into the plant genome. It can also determine the number of gene copies inserted, though it's more labor-intensive than PCR-based methods. Phenotypic analysis involves observing visible traits resulting from transgene expression, such as herbicide resistance or color changes. While it provides simple visual confirmation, not all transgenes produce visible phenotypes, so molecular methods are still essential. Molecular verification before proceeding with further research is absolutely critical. It prevents false positives, saves resources, and provides the validation required for publication and regulatory approval. Environmental conditions significantly impact both transformation efficiency and regeneration success in agrobacterium-mediated gene transfer. Temperature is a critical factor. The optimal range is 22 to 25 degrees Celsius. Higher temperatures promote bacterial overgrowth, while lower temperatures reduce virulence gene expression. Light conditions also play a key role. The standard 16-hour light, 8-hour dark cycle with moderate intensity provides optimal results. Higher light intensity can induce oxidative stress. Humidity should be maintained at 60 to 70%. Proper airflow and carbon dioxide levels also influence transformation success by affecting plant physiology. To summarize, the optimal environmental conditions include temperatures of 22 to 25 degrees Celsius, 16-hour light and 8-hour dark cycle, light intensity of 50 to 100 micromoles per square meter per second, and relative humidity of 60 to 70%. Deviations from optimal conditions can significantly reduce transformation efficiency. Temperature stress decreases tDNA transfer, light stress causes oxidative damage, and humidity fluctuations lead to tissue desiccation. For best results, use controlled growth chambers to maintain consistent conditions. Monitor parameters throughout the transformation process and adjust protocols based on your specific plant species requirements. Selecting the appropriate Agrobacterium strain is critical for successful plant transformation. Agrobacterium strains differ in several key properties that significantly affect transformation efficiency. These include virulence genes, host range specificity, antibiotic resistance markers, and chromosomal background. Let's examine the most commonly used Agrobacterium strains in plant biotechnology. GV3101, based on the PMP90Ti plasmid, has high virulence and broad host range, making it ideal for most dicots, including Arabidopsis. EHA105 features the PE105 plasmid with very high virulence and exceptionally broad host range, making it suitable for cereals and recalcitrant species. LBA4404, with the PAL4404 plasmid, has moderate virulence and more limited host range, but works well with solanaceous plants like tobacco and tomato. Virulence factors significantly impact transformation efficiency in different plant species. This chart shows transformation efficiency in Arabidopsis using different strains. EHA 105 delivers the highest efficiency at 85%, followed by GV 3101 at 65%, while LBA 4404 shows lower efficiency at 45%. Different agrobacterium strains exhibit varying levels of host specificity across plant families. For Brassicaceae species like Arabidopsis, GV 3101 and EHA 105 are preferred. Solanaceous plants like tobacco respond well to LBA 4404. EHA 105 excels with cereals and legumes, while recalcitrant woody plants often require highly virulent strains like AGL1. When selecting an agrobacterium strain for your experiment, consider these key factors. Consider your target plant species, desired efficiency, vector compatibility, selection markers, and your lab's experience with specific strains. Remember that a strain efficient for one species may fail with another. Always run pilot experiments with multiple strains when optimizing new transformation protocols. Agrobacterium-mediated genetic transformation has revolutionized crop improvement by enabling precise insertion of beneficial genes into plant genomes. Crop improvement through genetic engineering focuses on 4 key trait categories that address major agricultural challenges. Bee cotton contains genes from Bacillus thuringiensis bacteria that produce proteins toxic to specific insect pests like bollworm. This technology has reduced insecticide use by up to 80%, increased yields by 20 to 30%, and has been adopted in more than 15 countries worldwide. Herbicide-tolerant crops contain genes that allow them to survive application of specific herbicides that would normally kill the crop along with weeds. Popular examples include Roundup-Ready soybeans with glyphosate resistance and Liberty Link corn with glufosinate tolerance, both using bacterial genes to confer resistance. Drought-tolerant crops use genes that regulate stress response pathways to improve water use efficiency and yield stability. Commercial examples include drought-guard maize, which maintains yields with 30 to 50% less water, and experimental drought-tolerant wheat varieties being developed for arid regions. Golden rice is engineered to contain beta-carotene, a precursor of vitamin A in the rice endosperm. It contains genes from soil bacteria in either daffodil or maize to enable this biosynthesis pathway. This innovation has the potential to prevent 250,000 to 500,000 cases of childhood blindness annually in regions where vitamin A deficiency is prevalent. The impact of genetically engineered crops extends beyond individual traits, leading to increased yields, reduced chemical use, enhanced food security, and improved nutritional outcomes. Agrobacterium-mediated transformation has enabled plants to serve as biofactories for valuable compounds. This process, known as molecular farming, transforms plants into living factories that can produce pharmaceuticals and industrial compounds. In pharmaceutical applications, transformed plants can produce vaccines against diseases like hepatitis B and influenza. They can also produce antibodies for diagnostic and therapeutic use, as well as therapeutic proteins like insulin and growth factors. For industrial applications, plants can be engineered to produce enzymes like amylases and cellulases used in manufacturing processes. One of the most promising areas is the production of bioplastics and biodegradable polymers, addressing environmental concerns about plastic waste. Plants can also be engineered to produce specialty oils and biofuels, offering renewable alternatives to petroleum-based products. Plant-based production systems offer significant cost advantages over traditional production methods. Traditional biopharmaceutical production can be expensive, requiring complex fermentation or cell culture systems. In contrast, plant-based systems can reduce production costs by 50 to 90% through simpler growth requirements and scalability. Plants, as biofactories, offer a unique advantage. They can produce complex compounds through their natural growth processes. Once transformed, these plants can synthesize valuable compounds that accumulate in their tissues, ready for extraction and purification. While agrobacterium-mediated transformation is a powerful technique, it faces several limitations and challenges that researchers must overcome. Many plant species, particularly monocots and gymnosperms, are naturally resistant to agrobacterium infection, limiting its applicability. Even within the same species, different cultivars can show dramatically different transformation efficiencies, requiring optimization for each genotype. Agrobacterium-mediated transformation protocols are time-consuming and require specialized skills and equipment. There are physical limitations to the size of DNA that can be transferred, typically between 25 and 30 kilobases for binary vectors. Regulatory frameworks for genetically modified organisms create significant hurdles for commercial applications of this technology. As agrobacterium-mediated transformation continues to evolve, several emerging technologies are poised to revolutionize this cornerstone of plant biotechnology. Integration of CRISPR-Cas9 delivery through agrobacterium enables precise genome editing within the transformation process. This approach allows researchers to deliver editing machinery and make targeted genetic modifications with reduced off-target effects. Nanoparticle-assisted transformation represents another frontier where engineered particles enhance DNA delivery efficiency and protect genetic material from degradation. These approaches are especially promising for recalcitrant plant species that have traditionally been difficult to transform. Automated high-throughput systems are revolutionizing transformation processes through robotic tissue handling, standardized protocols, and artificial intelligence for optimization. These advancements dramatically increase throughput while improving reproducibility across experiments. As research continues, agrobacterium-mediated transformation expands to new plant species while becoming faster, more efficient, and cost-effective. The integration of these emerging technologies ensures this method will remain a cornerstone of plant biotechnology and play a critical role in developing sustainable agriculture solutions for our changing world.